In the previous lecture, we examined coral reefs, highly productive, structurally complex ecosystems that thrive in shallow, wet-lit environments through efficient nutrient recycling and symbiosis. Today, we shift to a very different marine environment, the open ocean, which covers the vast majority of Earth's surface. Unlike coral reefs, the open ocean is typically characterized by low nutrient availability, minimal physical structure, and strong dependence on large-scale ocean processes. Despite these constraints, it supports enormous amounts of life and plays a central role in global biogeochemical cycles. In this lecture, we will examine how physical processes, nutrient dynamics, and biological adaptations shape life in the open ocean, and how organisms survive and function in what is often a resource-limited environment. The open ocean is referred to as the pelagic environment, which includes all water away from the sea floor and coastal boundaries. Unlike coral reefs or coastal systems, the pelagic environment lacks fixed physical structure. There are no surfaces to attach to, no crevices for shelter, and no defined physical territories. This creates a fundamentally different set of constraints. Organisms are living in a three-dimensional moving fluid where they must constantly deal with sinking, with drag, and the challenge of locating food in what is often a dilute environment. As a result, pelagic organisms are shaped by three core functional demands: maintaining position in the water column, moving efficiently, and detecting prey. Start with buoyancy. Small phytoplankton cannot actively swim to maintain position, so they rely on physics. Diatoms, for example, increase drag with spines and elaborate silica structures, slowing their sinking rate. Some cyanobacteria use gas vesicles to regulate density and adjust their vertical position in response to light or nutrients. Zooplankton take a different approach. Many have bodies with very high water content and minimal dense tissue, making them nearly neutrally buoyant. Jellyfish are a good example. Over 95% water, so they neither sink rapidly nor require much energy to stay suspended. Larger organisms use more active control. Most bony fishes regulate buoyancy with a swim bladder, adjusting gas volume to maintain neutral buoyancy at a given depth. Sharks lack this structure and instead rely on large, lipid-rich livers to reduce their overall density. Next is locomotion, which is strongly constrained by density and viscosity of water. Fast pelagic predators like tuna or wahoo, like you see in this picture, have streamlined fusiform bodies that minimize drag and allow for efficient, sustained swimming. Their body design is optimized for continuous movement through open water where prey is patchy and often mobile. Detecting prey in a relatively featureless environment requires specialized sensory systems. Vision is critical in the open ocean. Many pelagic fishes have large eyes adapted for detecting contrast and motion in clear water. In deeper, dimmer environments, this shifts. Some mesopelagic fish have upward-facing tubular eyes that detect silhouettes against the faint light from above. Many predatory fish have countershading design that makes them difficult to see. Look at the photo on the right of this slide. If you look carefully, you'll see a great white shark as viewed from underneath. As you can see, they can approach very close to prey without being detected until it's too late. Where light is absent, organisms rely more on mechanosensory and chemical cues. Fish can detect pressure waves with their lateral line system, while sharks use highly sensitive olfaction to track prey over long distances. The pelagic zone is also vertically structured. The epipelagic zone receives enough light for photosynthesis, supporting phytoplankton and the food webs built on them. Below this, light declines rapidly through the mesopelagic and disappears entirely in deeper zones. These vertical gradients matter because the strategies we just discussed, buoyancy, locomotion, sensory systems, all of those things will shift with depth, reflecting changes in light, pressure, and resource availability. Light availability is one of the dominant controls on how the open ocean functions, but the key point is not just that light decreases with depth, it's how quickly and unevenly that decline happens. As sunlight enters seawater, it is both absorbed and scattered, and different wavelengths disappear at different rates. Longer wavelengths, like red, are lost within the first few meters, while blue light penetrates much, much deeper. That's why open ocean water appears blue and why organisms living at depth are often tuned to blue wavelength specifically. What matters biologically is that light intensity declines exponentially, not linearly. That means most of the usable light energy is concentrated very, very close to the surface, and even just a small change in depth can produce large changes in available light. Because of this, primary production is highly compressed vertically. Phytoplankton are operating within a relatively narrow depth range where light levels are sufficient to enough photosynthesis to keep them alive. Below that, photosynthesis cannot keep up with respiration, and they will die. As you know from previous lectures, this creates a strong vertical gradient in biological activity, where energy input into the system is concentrated near the surface and then redistributed downward through sinking organic matter, grazing, and vertical migration. This introduces an important concept. Productivity is not controlled by light alone, but by the balance between light availability and the time spent in the light. Even in regions with high surface light, strong mixing can suppress phytoplankton growth because the mixing pushes phytoplankton below the photic zone. So rather than thinking of light as simply present or absent, it's more accurate to think of it as a steep dynamic gradient that interacts with physics and behavior to structure where the organisms live, feed, and survive in the open ocean. One of the defining characteristics of much of the open ocean is nutrient limitation. As we discussed in the primary production lecture, phytoplankton requires nutrients such as nitrogen and phosphorus to grow. In the open ocean, these nutrients are often in very low concentrations at the surface. This pattern arises because phytoplankton continuously take up nutrients in the euphotic zone. Once incorporated into biomass, these nutrients are removed from the dissolved pool. When organisms die or produce waste, organic material sinks through the water column. As this material sinks, it is decomposed, releasing nutrients at depth. This process leads to a vertical separation between surface waters, which are nutrient poor, and deeper waters, which are nutrient rich. In many open ocean regions, the water column is strongly stratified, meaning that surface and deep waters do not mix easily. This prevents nutrient-rich deep water from reaching the surface, and that, of course, limits phytoplankton growth. Of course, these conditions are referred to as oligotrophic, meaning low in nutrients. We have talked about that before. The bottom line is that primary production remains limited because the necessary nutrients are not readily available, although sunlight is plentiful and very abundant. The vertical structure of the ocean is not uniform. It is organized into layers based on density differences, and these layers play a central role in controlling the nutrient availability and biological productivity in the open ocean. Density in seawater, as you know, is primarily determined by temperature and salinity. But in most open ocean settings, temperature is the dominant factor. Warm water is less dense than cold water. So when the surface ocean is heated by the sun, it forms a lighter layer that sits above the colder, denser water below. This creates a region of rapid temperature change with depth known as a thermocline. Within this layer, temperature decreases quickly over a relatively small depth range. Because temperature strongly influences density, this region often coincides with a sharp density gradient called the pycnocline. The pycnocline acts as a physical barrier to vertical mixing. Water above and below this layer does not easily mix because doing so would require moving denser water upward or lighter water downward, both of which are energetically unfavorable. As a result, the ocean becomes stratified with relatively stable layers. This stratification has major consequences for nutrient distribution. As we discussed earlier, nutrients are regenerated at depth through the decomposition of sinking organic matter. However, because the pycnocline limits mixing, these nutrients remain trapped below the surface layer. At the same time, phytoplankton in the surface ocean continue to take up available nutrients. Without a mechanism to resupply those nutrients from below, the surface layer becomes progressively nutrient depleted. Mixing events such as strong winds, seasonal cooling, or changes in circulation can weaken or disrupt stratification. When this happens, the pycnocline becomes less stable, allowing deeper, nutrient-rich water to move upward into the surface layer. This can stimulate phytoplankton growth and increase productivity. So the thermocline and pycnocline are not just physical features, they are key regulators of biological activity, controlling the extent to which nutrients can reach the sunlight surface ocean and support primary production. Primary production in the open ocean is not uniform. Instead, it varies significantly across different regions depending on nutrient availability and physical processes. The centers of subtropical gyres are among the most nutrient-poor regions of the ocean. These areas are strongly stratified and receive very little input of nutrients from deeper waters. As a result, phytoplankton productivity is relatively low. You can see that on the productivity map on the right-hand side of the slide. In contrast, regions near the edges of the gyres or along ocean boundaries often exhibit higher productivity. These areas may experience increased mixing, upwelling, or interactions with coastal processes that bring nutrients into surface waters. These patterns are ultimately controlled by how physical circulation redistributes water masses. In subtropical gyres, surface waters converge and sink slightly, reinforcing stratification and isolating surface waters from deeper, nutrient-rich layers. In contrast, at boundaries in equatorial regions, divergence or mixing processes bring deeper water upwards, supplying nutrients that stimulate phytoplankton growth. Equatorial regions can also show elevated productivity due to upwelling associated with wind-driven divergence of surface waters. This brings nutrient-rich deep water into the euphotic zone. These special patterns illustrate how physical ocean processes shape biological productivity on a global scale. Understanding these patterns is essential for interpreting marine ecosystems and global carbon cycling. Organisms in the open ocean must cope with environments where nutrients are scarce. As a result, many phytoplankton species have evolved adaptations that allow them to survive under these conditions. One key adaptation is small cell size. Smaller cells have a higher surface area to volume ratio, which enables their ability to absorb dissolved nutrients from the surrounding water. This gives them a competitive advantage in nutrient-poor environments. In addition to size, many open ocean phytoplankton have developed highly efficient nutrient uptake systems. These may include specialized transport proteins that allow cells to absorb nutrients even at very low concentrations. Growth strategies also differ from those in nutrient-rich environments. Instead of rapid growth during short periods of high nutrient availability, many open ocean species grow slowly but steadily, maintaining populations under stable, low nutrient conditions. As a result of these constraints, open ocean communities are often dominated by very small phytoplankton, such as picoplankton and nanoplankton, including groups like cyanobacteria. These organisms are highly efficient at nutrient uptake and are well suited to oligotrophic conditions, but they also influence the structure of the food web by favoring microbial pathways over direct grazing. One of the most widespread and ecologically important behaviors in the ocean is diel vertical migration, often abbreviated as DVM. This is a daily movement pattern in which organisms move up and down through the water column over a 24-hour cycle. It is not a minor phenomenon. This is considered the largest synchronized migration on Earth, involving billions to trillions of organisms across the global ocean, and it happens every single day. The basic pattern is consistent across many taxa, including zooplankton, small fish, and even some larger organisms. At night, these organisms migrate upward toward the surface, entering the upper ocean where phytoplankton and other food resources are most abundant. During the day, they descend into the deeper, darker waters. To understand why this occurs, we need to think in terms of trade-offs between feeding and predation risk. The surface ocean provides access to food, particularly phytoplankton, but it also creates a dangerous environment during daylight hours because many predators rely on vision to locate prey. In clear ocean waters, visibility can be quite high, making small organisms more vulnerable. By remaining at depth during the day, organisms reduce their visibility to predators. The deeper water is darker, and this provides a refuge from visually hunting predators such as fish. Then, under the cover of darkness, these organisms migrate upward to feed when predation risk is reduced. This behavior is tightly linked to light intensity, not just time of day. Many organisms respond to very small changes in light level at dawn and dusk, triggering their vertical movements. This indicates a finely tuned physiological response to environmental cues. Diel vertical migration also has significant implications beyond individual survival. Because these organisms feed at the surface and then descend to depth, they actively transport organic material downward through the water column. This process contributes to the movement of carbon and nutrients from surface waters to deep layers. In this way, DVM is not just a behavioral adaptation, it's a biological transport mechanism that links surface productivity to deep ocean processes. It represents a direct connection between organism behavior and large-scale ocean biogeochemistry. The biological pump is one of the most important processes linking biological activity in the surface ocean to the chemistry and structure of the deep ocean. It represents the movement of carbon and organic material from the surface to depth, driven by biological processes. To understand this, we need to connect several ideas from earlier in the course. In the surface ocean, phytoplankton performs photosynthesis, converting dissolved carbon dioxide into organic carbon. This is the entry point of carbon into the marine food web. As phytoplankton are consumed by zooplankton and other organisms, this carbon is transferred through the food web. Some of it is respired back into CO2, but a portion becomes part of organic material that can sink through the water column. This sinking occurs in several forms. Dead phytoplankton cells, fecal pellets produced by zooplankton, and aggregated particles known as marine snow all contribute to the downward flux of organic material. These particles vary in size and composition, but collectively they represent a continuous rain of organic carbon from the surface ocean to the deep sea. As these particles sink, they are subject to decomposition by bacteria and consumption by other organisms. This process releases nutrients and carbon dioxide back into the surrounding water, primarily at depth. This is why, as we discussed earlier, nutrient concentrations increase with depth in the ocean. However, not all of this material is remineralized in the upper water column. A fraction continues to sink in deeper layers and may eventually reach the sea floor. This represents a long-term transfer of carbon away from the atmosphere and ocean surface. The biological pump is therefore a critical component of the global carbon cycle. It helps regulate atmospheric CO2 levels by sequestering carbon in the deep ocean. Importantly, this process is not purely physical. It depends on biological activity. The rate of primary production, the structure of food webs, and behaviors such as diel vertical migration all influence how much carbon is exported to depth. This is where everything comes together. The biological pump integrates primary production, nutrient cycling, food web interactions, and the physical ocean structure and mixing. It's one of the clearest examples of how biological and physical oceanography are inseparable, and it highlights how processes at the microscopic scale can have global consequences. I consider this figure on the screen to be probably the best figure that summarizes everything that oceanography and marine biology is really about. I expect everyone to be able to understand everything that you see on this figure by the end of this course. The open ocean should be understood as an integrated system in which physical, chemical, and biological processes are tightly connected. Light availability defines where primary production can occur, while nutrient supply determines how much production is possible. Physical processes such as stratification, mixing, and circulation regulate the movement of nutrients and organisms within the ocean. These processes influence phytoplankton growth, which in turn shapes plankton dynamics and food web structure. Biological processes, including grazing, recycling, and sinking of organic material, further influence how energy and nutrients move through the system. These interactions extend beyond local scales. The open ocean is connected to coastal systems, to coral reefs, and to deep sea environments through the movement of water, organisms, and energy. What should become clear is that open ocean is not limited by a single factor, but by the interaction of multiple constraints. Light defines where production can occur, nutrients define how much production can occur, and physical structure determines whether those two conditions overlap. The biological responses we see, plankton size, food web structure, carbon export, they're all direct consequences of how these constraints interact. The system's perspective is essential as we move into the deep ocean where energy is no longer produced locally but must be transported from the surface. Today we examined how the open ocean functions as a large dynamic system shaped by the interaction of physical processes, nutrient availability, and biological adaptations. We saw how stratification, mixing, and circulation influence productivity and how organisms adapt to survive in nutrient-poor environments. We also explored how energy moves from the surface ocean to deeper water through processes such as the biological pump linking different parts of the ocean system. In the next lecture, and our final lecture, we will move into the deep ocean, where conditions change dramatically. We will examine how life persists in environments without sunlight and how hydrothermal vent systems support ecosystems based on entirely different sources of energy.